Good afternoon, good morning, or good evening, depending which part of the world you are. And welcome to the EUROTOX webinar. So it's one webinar, the series of webinars that EUROTOX is running. If it's first time for you being here, welcome, and I hope you can continue following the EUROTOX webinars. And for those of you who are familiar and who have been here before, welcome back. And for today, it gives me great pleasure to introduce to you Professor Nynke Kramer. Nynke has gained a BSc in Life Sciences from the University College of Utrecht, followed up by a master degree in Environmental Change and Management from the University of Oxford, and then carried on her studies to end up with a PhD in toxicology from the University of Utrecht. She's currently associate professor in toxicology at the University of Wageningen. So she moved from Utrecht, where she was previously assistant professor in toxicology, to now in Wageningen. This was in 2021. And her research focuses very much on enhancing and the uptake of in vitro models in chemical safety assessment, chemical risk assessment, by developing models that help extrapolating toxic concentrations in vitro cell assays to toxic doses relevant to human and animals. She has published over 70 peer-reviewed publications, and she's very much that work very much that cover very much the identification of physiological and chemical parameters that are important in doing this extrapolation from in vitro to in vivo. Nynke's research has been awarded with a CTAC Procter & Gamble Fellowship for doctoral research in environmental sciences, and she's also very much engaged in EUROTOX activities, where she's the board member of N2 Tox specialty section, and she's also a member of the scientific committee of Society of Toxicology in the US. So today she'll be sharing with us more of her work on this extrapolation from in vitro to in vivo. And the title of her talk is Physiologically Based Kinetic Modeling in Next Generation Risk Assessment. And enough from my side. Over to you, Nynke. Thank you very much for giving this presentation.
Thank you for that introduction. So yes, thank you, George, and thank you. It's an honor to be here today. Apologies for the little late start. As George has already said, so it's I hope you can see my screen. I would like to introduce physiologically based kinetic models in next generation risk assessment. So for those of you, I promise I won't show a lot of equations. I really keep that to a minimal, but really just to illustrate what we can do and what challenges we still face when implementing these type of models in a risk assessment framework. So the aim of this presentation today is to explain how PBK models are essential or is essential in next generation risk assessment. Now, I do realize that for those that are not familiar with this field, this can sound a bit like gibberish, as it did for me when I started this decades ago, well, maybe a decade ago. And I thought but I was so grasped by it because it's such a powerful tool, and particularly in the context of how we could use it to improve the safety of our chemical exposure. So this is in order to just implement the same seed as the seed that I was implemented in me, sown in me. I'm going to introduce next generation risk assessment and specifically quantitative in vitro to in vivo extrapolation. And from there, I hope to give you the basics of physiologically based modeling, and then describe some of the applications of these models in risk assessment, how we can understand this, the safety hazards, the safety risks of our exposure to chemicals, And then last but not least, still identify a couple of the challenges that we still have and where we can improve. So bear with me, but as many of you are obviously well aware, as a EUROTOX society, toxicity testing and regulatory toxicology means a lot of animal testing, from acute toxicity tests to subchronic tests to toxicokinetics tests, where we give the animal the chemical and then observe how fast it leaves the body, but also chronic tests, reproductive toxicity tests, oncogenicity tests, etc., etc. And this is how we've done our hazard assessments. And from there, we obtain dose response relationships. We obtain them in these animal tests. We generally assume that the average human is more sensitive, just to be on the safe side, compared to the animal tests, and then that the sensitive human is about 10 times more sensitive than the average human. And so from there, we derive a reference dose in the US or a health-based guidance value here in the EU. And that, where do these uncertainty factors come from? How do we assume these sensitivity differences when we know that different species react? One species is more sensitive than the others, that humans react differently to the same dose of a drug or a chemical. And so we apply these uncertainty factors to account for these possible differences, because we know that an animal and a human, they process the chemical differently. So the way we metabolize it can be faster or slower, but also that, the receptor to which a chemical binds and then causes a certain effect can be, that affinity can be different. So we account for this kinetic differences and these dynamic differences with this factor, combined factor of 10 if it's for non-genotoxic compounds, but it's not necessarily based on the science as we know it. But it is based on this notion that there is a difference between humans and animals and within humans. So the toxicodynamic differences, so those are the differences in the way that the chemical reacts in the body, so what the chemical does to the body, but also toxicokinetic differences, so how the chemical is absorbed into the body, is distributed in the body, if it's distributed to the target organ, like a brain in the case of mercury, for example, or away from the target organ, how it gets metabolized, if it's bioactivated, and to what extent, and then how it gets excreted, if it's through urine or through biliary excretion, or if it's the elimination half-life is very high, for example. So toxicokinetics here really means what the body does to the chemical. And that's an essential distinction to make, because PBK models really focus on this toxicokinetic processes, so the absorption, distribution, metabolism, and excretion. And so when we're talking about next generation risk assessment, is really when we want to move away from our reliance on animal-based toxicity testing in our risk assessment. At least that's also how I started coming into this field. So it's really replacing this whole suite of animal tests with some of the newer tools that we have, which include some of the in silico tools, but particularly also a lot of these in vitro tests. And the aim is very clear. We're trying to move away from these animal tests, so at least, reduce them to a minimum from an ethical point of view, but also from a scientific point of view, because it's still, an animal is a bit of a black box model. We assume it's similar, but with these uncertainty factors, we know and we indicate that it's not. But it's also, what it doesn't give us as much as we might like is this mechanistic understanding of why a chemical causes toxicity at a particular dose. And then related to that is the incredible costs that are associated with testing just a single chemical for its toxicity. So it's and given the number of chemicals that we're being exposed to, we've got a while to go. And so this is obviously also the framework of our Article 4 in Directive 2010 release here in the EU, and that feeds into the Cosmetics Act, for example. And the aim is to replace, or at least to reduce, refine, or replace these animal tests with, for example, in vitro toxicity assays. And this was coined back already in the landmark paper by the National Research Council in the US, Toxicity Testing in the 21st Century, where in vitro tests, so these are cell-based assays or subcellular assays where you can expose a plethora, lots of concentrations of a chemical in specific cells, so you can really get a mechanistic approach or understanding of the toxicity, and then it provides us with at least the basis of an ethically sound science. We can use human tissue, so we avoid that species extrapolation if we're interested in human toxicity, of course. And together with it, it can be done in a very high throughput manner with a little waste. And so, given that in vitro tests have been around for a while now, there are very few really that have been validated as an alternative to some of the toxicity tests that we have, at least on an OECD level. I check the internet, I check the OECD once in a while to see what's been updated. But these are just a handful, and it's a very slow process to start replacing that. And when thinking about it, it makes sense because, and I think this example of ToxCast in the US really exemplifies it well, where in vitro tests are just a small inkling, a small part of an entire body that drives toxicity. So when they screen, for example, in the US, the 10,000 environmental chemicals for which there is, or at least there is risk information missing, and they use these very high throughput robotic in vitro systems, and they screen them for all these possible disruptions in biological pathways. As you can see on this graph here, I'll give you a pointer. You can see there's a long list of predominantly pesticides here. This is the first of the 10,000 screens. But you can see that almost all chemicals seem to at least cluster around the same activity concentration or median effective concentration. So you've got rotenone here that is a little, that is very reactive. And so some assays have a very low AC50. But generally from here, it's hard to start prioritizing. And so what are we missing? Well, if we look at an animal test, it's an incomplete picture because we put something in the animal and out comes an effect, very crudely said. But with in vitro tests, we still have an incomplete picture because we now much better understand what happens at the target, so a molecular initiating event at a receptor in a particular tissue. But how does that translate to our exposure and to our risks? This is where we need more than just these in vitro assays. So this next generation risk assessment is really that combination of in vitro but also in silico approaches. And that is really to fill in the individual steps that lead to a toxic outcome. So going from a source to an external dose, like milligrams per kilogram body weight per day, to an internal dose like a plasma concentration, to a target dose like a concentration in the brain where the toxicity happens, and then the interaction at the brain, for example, at the acetylcholinesterase enzymes or neurotransmitter inhibition, which then leads to effects on the cellular level, on the tissue level, and then eventually an organism level. And so these, where what we need is a plethora of tests, and these include physiologically based kinetic models, and I'll show you how. So what in vitro tests can give us is a very good mechanistic understanding of what the chemical can do at the target site. The idea now is how much change at that target site. So for example, how much enzyme inhibition is needed to have a clinical outcome, or how much gene expression change is needed to have a clinical outcome. And this is where adverse outcome pathways and quantitative adverse outcome pathways really come in to really make that translation. So translating the y-axis of a concentration response curve in vitro to a dose response curve in vivo. But of course, that's one aspect of quantitative in vitro to in vivo extrapolation. What it's normally related to is actually understanding and translating the x-axis of a concentration response curve in vitro to a dose response curve in vivo. So that's going from a concentration in vitro so that's the target dose, to an external dose, so the dose in vivo. So that's understanding how a chemical goes from the tissue, how that relates to the concentration, for example, in plasma, and how that relates then to the external dose that we need to orally take in or inhale, for example. And this is really where physiologically based kinetic models, as well as in vitro distribution kinetic models, come in. So that's translating the x-axis. So that's the framework. Now into the first and the only differential equations or equations that I will really show you. So please don't worry. So physiologically based kinetic models are a series of mass balance differential equations as they simulate the transport of a chemical between tissues. So these models are really just a compartmental overview of how our body is structured. So every tissue is a compartment from which are connected to one another, usually by the bloods, by our circulation system. And so, if, for example, a chemical goes into the intestine, it moves into the liver through the portal blood, and then goes through the arterial blood, through the venous blood to the different and arterial blood to the different tissues, and that includes the target tissue, like something like the brain. And each of these compartments consists of a suite of equations, and I'll walk you through them very briefly. So the concentrate, and I'll start from the bottom just to make it confusing. So the concentration in venous blood in the brain, if that's the target organ, for example, is a function of the concentration that's actually in the brain and the partition coefficient, the blood-brain partition coefficient. So the extent to which a chemical likes sitting in the brain tissue compared to the blood tissue. And so what is the concentration in the brain? Well, that's the amount in the brain divided by the volume of the brain. And then what is the amount in the brain? Well, the amount of the brain is the integral of the change in the amount in the brain. And so what's the change in the amount of the brain? Well, that is a function of the blood flow rate to the brain and the blood flow rate out of the brain, and the concentration in the arterial blood to the brain and the concentration in the venous blood out of the brain. And so what is really neat about this system is physiologically based because of parameters like the blood flow rates and the volumes of tissues. And this makes it so powerful because it should be applicable to any system. So it's really not we do not, in that sense, need experimental data. In theory, it's a simulation. And what comes out when we each tissue has been given all these equations are these concentration time profiles of the chemical in each of the tissues that we've modeled here. So you'd see how a change or a dose decreases, for example, in the liver, or in the blood, but increases in a particular tissue. And the input parameters in these PBK models are threefold. The first is obviously the dose, the input dose, whether it's an external dose or an internal dose, but then also the organism properties like the blood flow rates and the volumes of tissue, but also importantly, chemical specific parameters. So these are parameters like the absorption, parameters that relate to the absorption. So for example, apparent permeability, which translates in the flux across the gut. The distribution parameters like tissue blood partition coefficients that I showed here earlier, but also the unbound fraction in blood. And then there's metabolism parameters, so how fast the chemical is metabolized, for example, by enzymes in the liver, so like cytochrome P450s. And this ideally is like a Km, Michaelis-Menten kinetics parameters like the Km, the Vmax, or the ratio of that's the clearance. And these are input into these differential equations. And then excretion parameters. Usually this is the glomerular filtration rate, but if there are transporters involved, we would also have some Michaelis-Menten kinetics. And because these are physiologically based, we could model, in theory, any species for which we have these physiological parameters. And so even in a nerdy mood, we've modeled the dinosaur. Not that we really had much true data to go there, but it just illustrates what the theoretical potential of these models are. The hardest part of a PBK model is really parameterizing them and finding the correct particularly, again, chemical specific parameters. But for this, we can use in vitro and in silico tools. So in vitro absorption, distribution, and metabolism models are generally well known. For example, the KCO2 permeability assay that you can see here describes how a chemical gets absorbed across the gut epithelial at a certain rate, which we can translate into flux and input into our PBK model, but also quantitative structure activity relationships, or quantitative property activity relationships, as you can see here by Joost de Jong, but there are a lot of them, where a chemical property, particularly the octanol water partition coefficient, correlates in a certain function with the blood, in this case fat blood partition coefficient. But we can also predict here the unbound fraction in plasma, for example, or the other blood plasma partition coefficients. So these parameters we can then obtain outside of the animal or the human. So this way we can really simulate. And this is why, since it's a simulation model and it's physiologically based, they have and can have many applications. This includes dose-dependent effects on kinetics, for example, where we can examine saturation effects of a drug. If we up the dose so much, what happens? And it gets into nonlinear kinetics, we could, in theory, predict it. And this is also what's done in pharmacology. We can also use it for species, to understand species sensitivity differences, at least on the toxicokinetic side. So understand why, from a toxicokinetic point of view, certain species are more sensitive to a chemical than others are. And of course, we can also understand, which is related to the first one, model additional exposure scenarios. When it comes to extrapolation, what we use it for in next generation risk assessment is really quantitative in vitro to vivo extrapolation, but also for high to low dose extrapolation, route to route extrapolation. So that, for example, we can use the model and we validated it for oral absorption. We can also then use it to understand how the kinetics and therefore the toxicity changes if we absorb the chemical through inhalation. But we can also then test sensitive individuals, just like we can test species differences. We can test what, for example, a patient with an impaired kidney, how they might react, or how the concentration of the chemical may change, and therefore the toxicity. And we can also use it to read across other chemicals with a similar structure, for example, and understand their kinetics and uptake and excretion, and so forth, etc. So this is where these have all been applications for PBK models, some more mature than others. What we're currently also working on a lot is in using these models for interpreting biomonitoring data between different locations and people. I wanted to give one classic example by my professor from here at Wageningen University, who's been working on these models for a while herself. So this is coumarin. Coumarin is a chemical in cinema, and that a number of you will probably be aware of, but is a hepatotoxicant. So it's found to cause liver tumors in rodents. And these tumors are associated with the formation of the bioactivation of coumarin to O-hydroxylphenol acetaldehyde that you can see here. So this is coumarin, and this epoxide here is thought to be associated with increased risk of tumor formation. And but there's the chemical itself also gets detoxified to 7-hydroxycoumarin by enzymes like cytochrome P452A6. So understanding the metabolism of the chemical can explain possibly why there are species differences. And it's true that in humans, this is the predominant pathway, whereas in rodents, it's the bioactivation pathway that drives the toxicity. And when using these PBK models, we can simulate exactly how much of that bioactive metabolite is being formed, and do this across species. And when doing this, for example, in rats, we can see, if we look at the units here and the numbers here, this metabolite is being formed at a standard dose here of 0.64 picomoles per gram of liver, whereas in the average human with cytochrome P453A4 activity and 2A6 activity, it is multiple orders below that level being formed in rats, and even in humans that are deficient in CYP2A6, those levels are not reached. So this explains why this uncertainty factor, that default uncertainty factor that we had initially applied, is not relevant for this particular compound, and we can actually set a chemical specific uncertainty factor. So instead of the uncertainty factor of 10, or at least 2.5 for change in the kinetics, we can remove that uncertainty factor altogether, which has been done in this case. And what this example also illustrates is how we can model different individuals which are poor metabolizers or ultra metabolizers, people that have a large body weight, a smaller body weight, etc. So these, instead of having a single value for each of these input parameters, like the Vmax or Km, we can also give it a distribution, and this is where most of the models are today. So this also gives you that actual or at least variability as opposed to uncertainty, at least in toxicokinetics. And what is really powerful is in this way we use it in at our institute, and the way we develop them is really understanding these in vitro effect concentrations and translating them to human bioequivalent doses or another species. So this is the first example of that by Joachim Louise, also in this lab a couple of years ago now, where he's was able to perfectly predict the dose response curves of the embryotoxic dose response curves of glycol ethers in rodents using a mouse embryonic stem cell test, where he obtained these concentration response relationships of the metabolite, and he set those concentrations that were toxic in vitro to the concentration equal to the concentration in the fetal blood, and he back extrapolated what the parent compound dose should be to reach these concentrations in the blood, and that gave, in this case, it's a beautiful example. It doesn't always look this good, but it gave a very nice dose response relationship. So using only in vitro assays in this PBK model, we could, in theory, in this sense, replace the animal test. And this is also what the US EPA has done in a slightly simpler model approach with HTTK, but it's a great example. So before, where all these in vitro effect concentrations didn't give a very clear example of which chemicals were more priority to start regulating than others. Once they use this quantitative in vitro to vivo extrapolation approach using a PBK model, we can see that there's that some chemicals indeed, as is known, are more hazardous than others, and some are more of a risk because of the level of hazard. So the toxic human bioequivalent dose is in the range of the expected exposure, and those are the green dots here. And so just a very quick reminder of how we set up these PBK models. So the steps, we follow approximately a 6-step approach. So we first, depending on the chemical and depending on the question that we're trying to answer, we develop a model. So we don't always model the entire, we don't have every single organ in the system in place, because that means we need all the parameters too, but only those that we deem important. So we have this, we decide the model structure, we translate them into a mathematical equations, then we define the input parameters, which is really the most time consuming. We solve the equations, we validate it with human or animal data that we have, and then we apply it in the context of quantitative in vitro to vivo extrapolation, or for chemical specific uncertainty factors or assessment factors. But of course, this is a very slow process. If we had to do this for every single compound, for every single application, but there are a number of generic PBK models out there. I've just given a list of the ones that we've worked with and we know well. Some are open source, others are very powerful, but not open source. But the majority that are out there we have put up here, and these include the US's EPA, HTTK, but also Indus Chemfate, which runs on Excel, which is a very neat, particularly for a starter, is a very neat way to introduce yourself to PBK models, but also ARVIS, Platham, EFSA, the European Food Safety Authority, has TK plate. And then we and Wageningen actually on Spoon set this up have the QIVIV tools, which is also a generic model, and I'll show you how it goes. But then also the big three, which I call the SimSip, or Satara's model, GastroPlus, and PKSim, which is very, they're well known for, particularly in pharmacology, and used there. These generic models have a set list of compartments of tissues, and where the user, so there's usually a user interface, the user would then input their chemical of interest, give physical chemical properties of the chemical, calculate using some of these QSARs, calculate these chemical specific parameters like the unbound fraction in blood or tissue blood partition coefficient, input parameters like the metabolic clearance parameters, or absorption parameters, and then there's some sort of play button where the outcome tends to be these concentration time profiles at the dose that was set by the individual. So these models are very powerful also and it also means you do not have to actually make these models yourself. And these generic models do quite well there. So there are a number of them. One I want to point out was prepared by René Cechi here as part of the EUROTOX project, and he prepared a high throughput PBK modeling approach and compared that model approach, this generic model, to actual measured human concentrations, C max concentrations in plasma and area under the curve. And in fact, for most of the compounds, the prediction was well done. So this shows you that for a lot of these chemicals, we can predict with these generic models. So 87% of the Cmax and 84% of the area in the curve was correctly at least predicted within tenfold of the experimental data. And this approach, it also means it's being picked up also from a regulatory point of view. The one that I've been involved with a lot of people, very smart people at EFSA, was to design a quantitative in vitro to in vivo approach using these PBK models for the D&T IVB, so that's developmental neurotoxicity in vitro battery, where the in vitro assays, we want to replace the D&T, the in vivo D&T assay with these 17 plus in vitro assays, but then we have to translate it obviously to a human or a rodent dose. And this paper I thought is very neat. Also shows that we can do this also for these very specific assays. So Anna Kreutz and colleagues have been comparing 3 of these generic PBK models, GastroPlus, HTTK, and PKSim, to derive the developmental neurotoxic dose of a number of compounds for which they actually had toxicity data, in vivo toxicity data, to calculate both the hazard, so in this case, the administered equivalent dose or the human bioequivalent dose of each of these D&T compounds, and then compare that to the exposure to understand the risk. For example, and the risk in this case can be exemplified by the bioactivity exposure ratio, or BER. And there it gives us a prioritization, which compounds could indeed be D&Ts, and D&T compounds that could possibly be at risk because the margin of safety isn't sufficient enough. Or also, what it also shows here is that 78% 8% of all the chemicals were actually predicted within threefold. So the effect, the toxic dose in vivo was predicted within threefold. And it also showed that there's not that much difference between the different generic model types. There are differences, and I won't go into this, but the differences are relatively small. So, which is very helpful from a perspective of user's perspective. And so this is also partly now this information, this type of modeling approach and the assessment of it has been summarized now in the EFSA's scientific opinion on the application of physiologically based kinetic models for quantitative in vitro to in vivo extrapolation, or QIYV, of developmental neurotoxicity. So I suggest that you do have a look at that paper. It also, what's very important, it also explains some of the challenges when setting up this opinion. There's not a lot of data to prove it yet, but there were a couple of challenges that we ran into, and some of this include that our simulation of particularly lipophilic compounds that could be improved for sure. That's where we see the largest differences in predicted and observed kinetic output. Then, of course, also the toxicokinetics of chemicals undergoing at extrahepatic metabolism is often not caught or simulated well in these generic PBK models. Also, chemicals that are transporter substrates. So substrates, for example, the organic anion transporter, and also understanding the differences in kinetics of isomers is often not always taken into account. For example, if a cis or a trans permethrin has a big influence on its half-life in the body. These are still the things that we still need to tweak out when it comes to modeling chemicals. And of course, what is also still a challenge and is being worked on is understanding the change in the kinetic profiles of chemicals across life stages. One of the largest challenges we ran into also when using these, when deriving this scientific opinion, was to decide on what in vitro dose metric and what in vivo dose metric to compare. So do we compare the nominal concentration in an in vitro system with the concentration in the blood of a particular tissue, or should we compare it to the tissue concentration, so it concentrates in the brain itself? That is still an ongoing debate. And of course, the idea is to connect. So with PBK models, we only look at toxicokinetics, but we don't link it directly to differences in toxicodynamics, for example, connecting it to quantitative adverse outcome pathways or to other toxicodynamic models. And of course, the idea now is to, how do we deal with, where we have very data poor chemicals for parameterizing these PBK models? That's another challenge. And of course, really quantifying the uncertainty in PBK model predictions is still one that we're working on and that we need to still grasp properly. And this is just, and then I won't keep you too long, but this is just one of those examples. That's okay. So now we know that there are a number of challenges that still need to be addressed. And one of them is, particularly understanding the chemical and biological, but particularly chemical applicability domain of a number of these in silico tools, but also in vitro tools to parameterize PBK models. So what you see here are 4 parameters in a PBK model, like the apparent permeability, and the common assays. Those are the ones that are in bold, used to parameterize, to find that parameter, to find the value for that parameter. So for example, for the unbound fraction, we often use rapid equilibrium dialysis, but rapid equilibrium dialysis doesn't work well for very lipophilic compounds, for example. And another aspect is for a number of chemicals, something like the unbound fraction in plasma, it can be predicted with the optimal water partition coefficient, but for chemicals like the perfluorinated alkylated substances, that is not a good parameter because it's a surfactant, so it sits at the interface of oils and fats. So the octanol water partition coefficient isn't a great parameter to understand its distribution. So with PFAS, for example, we're developing and have developed alternative methods to assess the unbound fraction in plasma. For example, Fabian Fischer in this case used solid phase micro extraction instead of rapid equilibrium dialysis to really understand the partitioning of PFAS between different components in blood, so albumin, lipids, storage lipids, etc. And from there, derive new dose quantitative structure activity relationships that we can then input in the PBK models. And we can also use, in the case of PFAS, the normal models are not mechanistic or not as mechanistic as we want because they don't capture, for example, the transport mechanisms of transporter activity that drives the kinetics of perfluorinated alkylated substances. So at Wageningen Food Safety Research, they've gone ahead and measured the Vmax and the Km for a suite of PFAS compounds, substances for OET4, which reabsorbs the chemical back from the filtrate into the blood, explaining some of the half-life, the long half-lives of these compounds. And so we prepared, and Crisanti did this, a PhD student in our lab has created a PFAS model, which is mechanistic, where we input these transport kinetics in there, as well as these new quantitative structure activity relationships for partitioning in between tissues and blood. And she was this is perfluorinated alkylated octanoic acid. You can see that she was able to predict experimental data and also use it to explain human biomonitoring data for these compounds. So this is an exciting development. So we can, even for these chemicals outside the applicability domain, we can start using PBK models for risk assessment and explaining human biomonitoring data. But I do want to highlight one of the other challenges is that, understanding what dose metric to relate. So what in vitro dose should be connected or linked to the in vivo dose to do that back extrapolation to an external dose. And this Sanana Poinka from a postdoc in our lab and now at SQ Labs, she's done a great study with colleagues in Switzerland, understanding amiodarone, which is a very lipophilic compound in these brain spheres, and looking at the neurotoxicity of amiodarone, but also showing that the nominal concentration, the concentration that we add to the in vitro system, doesn't sufficiently explain the variability in the outcomes based on the dosing regimen or the observed effects that we see thereof. And she also shows, and this is data from her, that with amiodarone, depending on which in vitro dose metric you use, whether it's the nominal concentration or the concentration in the cell, and whether you compare the concentration in the cell with the concentration in the fetal brain versus the nominal concentration in the blood concentration, you can have orders of magnitude different predictions of the human bioequivalent dose. So in conclusion, what I want to give the audience here is that what I think and we think here that PBK models are incredibly powerful tools, particularly in the context of next generation risk assessments, because they are a series of mass balance equations that simulate the transport of chemicals between tissues, and they are models that translate in vitro effect concentrations of human bioequivalent doses using other tools like QSARs or in vitro tools and therefore we can use it for quantitative in vitro to in vivo extrapolation. And therefore, what we do say though, because PBK models is a model using other models to parameterize it, it's really important to have a very good understanding of the biological applicability domains and the chemical applicability domains of the input parameters and of the models themselves. to understand and to start characterizing the uncertainties surrounding the predictions using these PBK models. And so it's another aspect here is to consider the in vitro and what in vitro dose and what in vivo dose metric should be used to compare, because they can greatly affect the outcome. And with that, I would like to acknowledge that this is not work that has all been done by me. In fact, it's been done by entire teams and great projects. So a lot of EU projects like ONTOX and PARC, but also the Dutch funding agencies. For example, the VHV for Safety projects. We've been working on these PBK models and their application in risk assessment, particularly also at the European Food Safety Authority. So I've lifted them here, and these are the wonderful people that have been doing a lot of this work. With that, this is my last slide. I'd like to thank you for your attention, and please, yes, I'm very open to answering any questions.
Oh, thank you very much, Nynke. I must put my camera on for a great overview about PBVK and really the challenges, but also the opportunities we have to work out going from external dose to internal dose right at the cellular level, and then back again from what we see at the cellular level into what would be a corresponding external dose. So that's fantastic. Thank you very, very much. We have time for some questions, and let me scroll up here. So I'll start with the first question from John Anto. What would you say is the strength of PBK in the practice of clinical toxicology?
Yes, in the practice of clinical tox, that's a great question. So I'm not involved in the clinic. But I know the clinical, it's actually been used there. So from clinical toxicology, particularly with some of those drugs that have a very low, a very narrow therapeutic range, this is where these models can be particularly helpful to assess, to adjust the dose of a drug. To be effective and maintain it within the range that is not toxic. I think this is one of the most, or other things like particularly drug-drug interactions. It's a very powerful tool there too.
Thank you. And then next question from Jonna Weissel-Leitinen. Would it be better to use specific PBPK models if working with risk assessment of certain chemicals, and are generic models better to use for screening prioritization?
Oh, that's yeah, it depends. So I didn't have time to show, but I would. It's like, so I know that at Unilever and collaborators, they've got a nice tiered approach. So the idea is that generic models, depending on the level of certainty that you need. So for example, the exposure versus the effect, I would say generic, you start with generic and you go to bespoke, unless there's already a bespoke model out there that you can, of course, use that. But I wouldn't say that you cannot use generic models for very specific questions. You could, but it depends very much on your margin of the level of uncertainty you're willing to consider.
Thank you. Then next question by Wolf, somebody called Wolf and Wolf. Question is it relevant to consider the average dose calculated by different models, like in benchmark dose modeling?
Oh, that is a good question.
Yes.
Yeah, is it relevant to consider the average dose? Yeah, so I guess with the same uncertainties with benchmark dose modeling, I don't know of any initiative where we've done that, so it's something to to consider. I'm not sure, but yes, I think it would be. So sometimes it would be relevant to check several assumptions, whether it's from the same model with different assumptions or different models, to see how the outcomes vary. But also, I would also try to understand why they vary, because it might be that the average is not good enough, but a very specific one is good enough. Yeah.
Okay, thank you. And we have two more questions. So the next one is, what are the current opportunities and applications for integrating toxicogenomics data into PBPK modeling to enhance mechanistic insight and next generation risk assessment?
Could you repeat that, George?
Yeah.
I tried to catch
What are
that.
the current opportunities and applications for integrating toxicogenomics data into PBPK modeling?
Yeah, so that's also again there are a number of people that are working on this. So I know it's ongoing research, and I think there is an opportunity, particularly. The most simplest form of that is understanding the genomics of some of these biotransformation enzymes and incorporating that. So of course, expression is not. Oh, sorry. Uh, a gene being there or not, it doesn't necessarily completely translate into an activity, but it's a good indicative way, particularly across species. It's a nice way to check. Yeah.
Okay, thank you. And last question. Do you think, it's also by Mr. Miss Wolf, do you think that reverse dosimetry can be used to define a NAM-based point of departure for risk assessment in cosmetics?
Yes.
Part of the next question, part of the question is, has this method gained sufficient regulatory acceptance to be used for regulatory purpose?
It's a so the short answer is yes for the first half of the question. The second short answer to the second half of the question is no. Um, but I think yes. So particularly in cosmetics, there's quite some good work by a number of people. The ones that pop to mind here are Unilever, which has really been looking at setting up this approach using for deriving NAM-based points of departure for risk assessment and illustrating how, and incorporating the uncertainties and how to use that in a tiered approach for risk assessment. So I think the framework's there. Now it's a question of illustrating its worth to regulators. But yes, I think so. I think it's very doable.
Okay, thank you very much. I don't see any more questions. And it also past 2 o'clock. So again, thank you very much, Nynke, for fantastic presentation and fantastic work. And thank you also to the audience for your participation, for the questions. And we'll see you in at the next webinar, which will be about a month's time. But keep an eye on the announcement. Thank you very much and goodbye.
Thank you so much. Thank you.